with genes, DNA, and you're good. You're good. Paying attention. I like that. All right, so lesson 2.3, genes, DNA, and cell.
Anderson Schmidt, please return to class.
Should I send a message and have them do a highly embarrassing Kartik and
Yes.
no, I can't be bothered. The joke will be on them when they don't know this and it's on the test.
Is there any paper I can steal from?
I don't have any at the moment, but I'm sure one of your fine fellow friends would be delighted to lend you some paper. Look at that. Look at this fine fella stepping up. All right, so as we said, all of the many traits
Wow.
a lot to unpack there, but that's okay. All of the many traits controlled by our genes can be found in the DNA of our cells. Okay, so let's remind ourselves. So for example, an animal cell. All right, let's imagine we've got a cell here. There it is. Inside the cell, we have the nucleus. Oh. Get some cytoplasm happening. Look at all that cytoplasm. I was never very good at coloring inside the lines. But that's okay. Will you forgive me?
Yep.
It's very kind of you. Oh, that was a bad one. Okay, well that's not very impressive, but that's okay. So, if that's the cell membrane, what do we call the fluid inside the cell? Cytoplasm, good. Cyto meaning cell, plasm meaning liquid. It's the cell liquid. This whole structure would be called the nucleus. Does anyone know what we call the membrane that surrounds the nucleus? The nuclear membrane. Does anyone know what we call the fluid inside the nucleus? Very good. And then obviously, what do we call this structure that is like DNA all coiled up? This would be at the time of the DNA reproducing, but that's okay. What do we call the actual structure of DNA? We call it a... Very good, a chromosome. Does anyone remember that? So if we look in the nucleus, let's imagine here we are. We'll zoom in a little bit. We'll take a few notes. In the nucleus, we have chromosomes which are long, very long, strands of DNA. How many chromosomes does a human have? No, I mean, I drew 2 because I didn't want to draw the real number. Yeah. You're close. Switch the numbers. Yeah. Humans have 23 unique chromosomes. And they're numbered 1 to 22, and then we do something weird with the last one, but we'll get to that in a minute. But again, we said, given that there's 23 unique chromosomes, each of these contains many genes that code for many traits. So, like somewhere on one of your chromosomes, the weird emphasis on a strange syllable. Parabola, that's right. So I want to show you, for example, spelling bee, that's not what I want to show you. For example, yeah, so like we talked about the hitchhiker's thumb, right, or the idea of that bendy thumb. And it's controlled by a gene, right? So that's not the gene I wanted. But anyway, let's take the gene that they did give here, which this is a different one actually, but that's okay. Just I want to show you for any gene imaginable. It doesn't matter. So here we have this gene called the SLC26A2 gene. These have very sort of hyper-specific names. If you go look at one of these, you can see where it's located, on what chromosome, everything like that. So if we go to its Wikipedia page, for example, what I want you to see here is that you can get all kinds of information about it, including where it's located. So for example, this particular gene, which, okay, ready? The sulfate transport is a solute carrier family protein that in humans is encoded by the SLC26A2 gene. It's also called the diastrophic dysplasia sulfate transport. Do we care about any of that? Here's the point. It's one gene that does one thing inside your cells. It moves sulfate ions. Not that you need to know that. Does that make sense? Here's the point. That gene, that little sulfate transporter that has to be built for your cells, there has to be a blueprint for it. Where's that blueprint located? It's located on chromosome number 5. And then on chromosome number 5, specifically it's located way the heck at this location here. Does that make sense? So, by the way, do you see how we've numbered the chromosomes? It's a pretty logical system. They've basically been numbered from biggest to smallest. And then we'll talk about these last little bits here in a sec. Does that make sense? So the biggest chromosome we call chromosome number 1. And then, they kind of progressively get shorter and shorter. So for example, chromosome number 5, which is where this gene is found, has all this here. There's chromosome 5. That's a real picture of a real one. And do you see how it has this kind of stripy pattern? When it coils up, it just kind of takes on that pattern. Protein number 5, chromosome number 5 is a fairly small one, where it's got, depending on how you count, anywhere from, we could say, about 800 to about 900 genes that code for proteins, and then about 900 more, or about 1,000 more genes that don't code for things, and then another 780 what we call pseudogenes. That's just on one of your chromosomes. How many chromosomes do we have? 23, right? So we could look at chromosome 1, for example. Given that it's longer, we'd probably expect it to have even more genes, right? And sure enough, oh, hello. No, Number 23. Well, 23 is interesting. We'll get to that. But there's chromosome 1. He's a handsome-looking guy, isn't he? Good-looking chromosome. And chromosome 1 has roughly 2,000 coding genes, 1,800 more non-coding genes, and about 1,400 more pseudogenes. We'll talk about what each of those things mean. Long story short, you've got a lot of genes. Does that make sense? And that makes sense because you have a lot of traits. Does that seem pretty logical? So, and we have the human genome fully mapped at this point, which means that we actually know what genes are in what location on each of your chromosomes. That only happened in the last 30 years. When I was in high school, they were still working on the Human Genome Project. Are you writing this down? Why don't you get a proper piece of paper? Yeah, okay, get on that. So, for example, I'm not going to draw all 23. We'll call that chromosome 1, 2, right, 3, ... 22. And then we'll talk about this in a minute. Not quite as big as chromosome 1. Really, 22 should be quite a bit smaller. Why it's very small. So, for example, there could be a single gene located here. Does that make sense? And again, what are the chromosomes made of? Sorry? DNA. Very good. Okay. So we could say here, example, locating a single gene. On our chromosomes. Now, not all animals have 23 chromosomes. But of course, we said you have 23 chromosomes, but what else do we know? We get our DNA twice. We get it once from your paternal, in other words, your father, and you get one from your maternal lineage, which is, of course, your mother. So we actually have 2 pairs of 23 chromosomes. Oh, whoa. For a total of how many? "23 from mom, 23 from dad."
27.
46 it is. Okay. Does that make sense?
Yeah.
Okay. So knowing that,
Shouldn't that be contained?
what did I say?
You said contains.
Each of these contains many genes. Yeah, because each is singular. The word implied after each is what? Each one of these, right? So each is singular, even though you're talking about 23 things. You'd say they contain, but you'd say each one contains. Does that make sense?
Yeah.
Cool. It's like how none is singular. We never say this. You actually should say like none is, not none are. Like none of them is coming tonight. But we don't really say that because we're not used to it, but that's correct, right? None is literally a contraction of what 2 words? No, one. So none should always be conjugated with a singular, but we almost never see that. But that is actually technically more correct. That's how I was taught. But if you talk that way, people look at you like you think you're special. You think some kind of fancy pants. None of them is. Anyway. All right. Do you need a sec or can I move on? We're good? Okay. So, That's a good question, isn't it? What is DNA?
Something that's inside.
Sure, potentially.
A buildup of your traits.
Ah, that's what's on DNA. Does that make sense? In other words, if I said, like, what's a book, and you said, well, it tells a story about blah, 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 that's true too, and that's absolutely true. But the other answer to what is a book is it's a bunch of pieces of paper bound together at the edge with a cover, and you flip between them. Does that make sense? And they're both true answers. So you gave one of the true answers about what's DNA. One answer to what DNA is it's a set of instructions and traits that tell the cell how to build a cell and operate a cell and run a cell. True. But the other thing DNA is like the actual physical thing itself, right? And what's that? It's a molecule, right? So let's talk about it. DNA is a large polymer. What does poly mean as a prefix? Maybe. But usually, like polygamous, somebody who's polygamous is married to multiple people. Somebody who's polyamorous is somebody who isn't married to, but is amorous or romantic with multiple people. A polygon is more than one gon, which means it's shapes with a bunch of sides. What else? Poly pocket has many pockets. No, that's not what that means. Anyway, so this is chains of smaller molecules making a bigger molecule. So DNA is like links in a chain. Does that make sense? Cool. So what's it look like? Each link is called a nucleotide. We actually discovered DNA thanks to viruses. See, the cell is big and it's complicated and there's lots of stuff in it. And so it was really hard to know what in the cell might be the genetic material. Do you know what I mean? Like if you take a human cell apart, there's so much stuff in it. And even if you take a bacterial cell apart, there's so much stuff in it. There's enzymes and there's...
Interruption.
Yeah.
Can I get your attendance, please?
Yep.
Thank you.
So there's DNA and there's RNA and there's proteins and there's enzymes and there's all kinds of things inside of a cell that it was really hard for scientists to figure out what in the cell is holding the information. Does that make sense? Why did viruses make it easy? Viruses are tiny and they're really simple. What are the two parts of a virus? You remember this? Yeah.
The protein capsule, which has DNA/RNA inside.
Cool.
And
that's it. So it's a protein capsule and DNA. So with a virus, and we knew that it passed new instructions onto the cell, right? So with a virus, we knew that the genetic material, the information must be stored either, because we didn't have a microscope that could see viruses, but we had taken them apart before, and we'd done chemical tests, and we knew that a virus is made of just two things, a slab of protein and a slab of DNA. And so the question then is, which one of those holds the information? And so scientists didn't know. So you know what they did? It's actually a famous experiment. It's a great experiment. How would you figure out? We know that viruses attach to cells, and we know that they pass new instructions on to cells. Well, what they did was they used radiation, where they made viruses where the DNA contained phosphorus, which the protein mostly doesn't. And so the DNA, they had it made from phosphorus that was radioactive. And then After the viruses had infected a bunch of cells, they washed it off very carefully. They washed it away, let the viruses grow, the cells grow and procreate, and then they checked to see if that radioactivity was inside the cells or if it had been washed away. And what they found was that it was inside the cells, thus proving that the DNA is the part of the virus that got inserted into the cell. Whereas when they marked the protein and made it radioactive, it didn't get put inside the cells. And that's how we first figured out that it's not proteins that carry genetic information, but DNA. We've done a lot of experiments since we know how this really works. After that, it wasn't that much later that they actually figured out what DNA looks like. So like we said, it's a big, long chain that is made up of links. And each link we call a nucleotide. And each nucleotide has the same structure. And it looks like this. Each one is made from a deoxyribose sugar. Deoxyribose sugar. We'll talk about that in a moment. A phosphate group. And what we call a nitrogen base or a nitrogenous base. Write that down and we'll draw it. We draw this as a little pentagon because it actually is pentagonal in shape. This deoxyribose sugar, which is a molecule made of carbon, hydrogen, and oxygen. That makes this little shape here. Then we have the phosphate group. Yep.
The first word, is that a B next to the I? I'm sorry.
Yes, I'm sorry, it is. We have our phosphate group here. Don't ever be sorry if my spelling is too messy to be read. That's not your fault, that's mine. And then finally we have, oh, hello. That's going to be too dark. My goodness. And then we have a nitrogen base. Critton is here. Evan is here. No Deanna. Saphira is here. No Audrey. Kayla's here. Aiden's here. Kira's here. Kartik's here. No Andrea. No Angela. No Annie. Max is here. Sam is here. Sam's here, right? Yeah, Sam's here. I don't know what just happened there. Cashew's here, Solange is here. Okay, there we go. Okay. Yeah. Did I do that?
I don't know if you did that.
The late slip should update that, so I don't know. I don't know what to tell you. And maybe. Yeah, she might not know what she's doing. Well, that's okay. Should go to the office and just tell them. So there we have this. So now what is DNA? So these nucleotides get stuck together in a chain. Oh, sorry. Okay, that's fine. We can put that there. That's okay. It doesn't hurt. That's not what I wanted to write next, though. I got my cheat sheet here. The phosphate and the sugar are always the same. But there are 4 different bases. And those are a molecule called adenine, they're called guanine, cytosine, and thymine. All right. Okay, we're almost there. So these are used as an alphabet to write code in DNA in the form of codons.
Why do you write these are used an alphabet?
As. Sorry.
Oh. Oh, I just instinctively.
So for example, if we looked inside of a cell, inside of the nucleus, at a section of DNA, right like this, and we looked at one particular section of it like that and zoomed in, what we would find is that the DNA actually is like G, C, C, A, G. So it's a base with guanine, then one with cytosine, cytosine, adenine, guanine, thymine, thymine, adenine, guanine, cytosine, dot, dot, dot. It's a big long chain, and the way this gets read is that these each form, oh, golly. Each 3-letter pair forms what we call a codon. Well, it's no different than how we write words, right? So if you want to say something, you put words together in a way that says that thing, right? So every word I'm using right now could be put in a different order and it would mean something totally different, right? So the order of the codons are used by the cell to tell it how to put together proteins. If you take the grade 12 university chemistry, we'll learn about how that works specifically, like really right down to the molecule how it works, and it's super interesting. But for the time being, enough to know that this says, okay, the next block for the protein is going to be this one. And the 3-letter code is enough to tell the difference between different codons. If you think about how many letters do we have in our alphabet?
We said
Yeah, so we can make a lot of different words, right? A 3-letter word could be a lot of different combinations, right? How big is this alphabet? Only 4 letters, right? Does that make sense? But at the same time, how many codes can you make from that? 3. So we have 3 possible combinations of 4. You don't have to write this, but the first one could either be A, G, C, or T. Then the second one could be either adenine, guanine, cytosine, and then the third one could be either adenine, guanine, cytosine. How many possible combinations is that? Well, there's 4 possibilities here, and then for each of those, it could go A, or it could be A, G, or it could be A, C, so on and so forth, right? How many combinations do you end up with? 4 possibilities here. How many possibilities here? And then how many possibilities here? 4, for a total of how many possible combinations? There are 4 times 4 times 4 equals 4 times 4 is 16 times 4 is 64. There are 64 possible codon combinations. And proteins are actually made from only 20 different building blocks. This code is able to, this code is more than enough for that. Does that make sense? The really crazy thing is all of the complicated things your body can do, right? You have muscles that can move and eyes that can see and vocal cords that can make sound and brains that can think, and all that stuff that your body does is all made of protein. Your brain is protein that does all that. Your muscles are protein. All the complicated, all your organs, all of that's all protein. And the crazy thing is that all of that stuff is made with only 20 different building blocks. We call them amino acids. Again, if you take the 4U chemistry class, you'll learn all about that. We just mentioned it briefly here, but only 20 building blocks. But all that complicated stuff. And your DNA just gives the instructions on how they get put together. Does that make sense? So the cell can look at the DNA, it can read that code, and it can use that to put together amino acids to make the building blocks of protein, to make these proteins that then can do complicated stuff. Any questions about that? That's pretty heady stuff. A little bit confusing? A little bit, right? But it doesn't need to be that confusing. I'll say this. If you want to cook something at home, you need to get I don't know, say you want to make chicken fingers. What do you need to do?
Chicken.
A bad example. Say you need to make macaroni. How do you make macaroni? You get the macaroni, you get the cheese, you get all that stuff. What do? You get a pot, right? And then you put water in the pot, you put it on the stove, you boil it, salt it a bit, boil it, then you put the noodles in, right? Then after they've cooked long enough, you take it off, drain the water, you put the cheese and the milk in, right? And a bit of butter sometimes, you mix it all up, then you eat it, right? Yeah. Okay. The box tells you how to do that, right? But weirdly, and maybe now, does that have pictures on it now? It didn't used to have pictures on it. It used to, I don't know. It used to just have words, right? Further up. How's that? Cool. Right? And if you think about it, how in the heck does a bunch of little squiggly shapes on there tell you how to make macaroni? But the answer is that those squiggly shapes we know make sounds, right? And then we know when they put together, they tell you a word, and we know the order to put those in. That's actually much more complicated than this. Because this is way more like, you know the instructions that come when you get Lego? Mm-hmm. You've seen those before, right? It says, get this block, put it here. Then get this block, put it there. This block, put it there. This block, put it there. Does that make sense? That's all the DNA is. The DNA just says, okay, you want to make this protein? Cool. Start with this building block. Next, you're going to add this building block. And so you literally have a machine inside your cell that moves along your DNA and reads those instructions and puts proteins together and builds them. That's it. It's actually pretty cool. So you have a factory inside every one of your cells that is able to read a set of instructions from this molecule called DNA and use that to build proteins. Kind of cool? I think it's kind of incredible, actually. We're going to leave it there for today because, again, boy, is it a tired Friday. Feels like a tired Friday to me.
It was pretty relaxing.
Yeah,
Could you scroll up a bit for me?
I would love to. Where to? Here?
Oh, that's perfect where I need.
Oh, look at me. Boy.
Amazing.
Ha! I'll take it. Oh. Yeah, you guys should be good to go. If they haven't set, let me know. We might just have to start fresh. But usually if you reheat it like that, it goes just fine. Did everyone get the names on those? And where did we leave them?
I put my initials.
Where did we leave them? Like on the table in there? Perfect. I'll probably move them next to my computer in the office and leave my computer on because that throws a lot of heat. And then it'll keep them at least a little warm. That's my thinking.
The entire weekend?
Solange, I like your shoes. Oh, yeah.
Isn't it bad for your computer?
No. We're fine. It's tough. It's like me, it's tough. First thing people notice about me.
How long does it take?
Bacteria? Well, they're reproducing right now. But how long will it take till you can see them?
9 months.
There have to be, no. A few days. But there have to be billions of bacteria before they're a big enough spot that you'd be able to see it with your naked eye. So they need to reproduce. Now the thing about that is, one bacterium, once it splits, will become 2, which when it splits will become then
8.
Great. So let's say this takes 10 minutes every time. How long will it take before you have a billion?
Not long.
What's not long?
Well, a billion.
Billion's a lot. So let's say we have time, and then let's say we have number of bacteria, right? And let's say it takes 10 minutes every time, right? Oh. Okay, there we go. I don't know how long this is going to take, so I'm assuming, fairly long. There we go. Okay, so we start with 1, and then what are we doing every time?
2.
We're taking the one above and we're doubling it, right?
Yeah.
So 4, 8, 16, 32, 64, 128, 256.
Wow.
Wow, that's
Right? And before you know it, that's quite a bit, right? How much are we there? That's 1 million,
So 1,000,000 at 200.
Right.
So probably.
and then before you know it, you're at 1 billion. Now that's assuming 10 minutes. I don't know if it's 10 minutes. I wonder what the average time for bacterial reproduction is. I suspect for most it's probably in the order of like hours, not minutes. Oh, it's hello. It's being weird. It's being weird. Okay. God, stop. Average time for bacteria to reproduce. There's going to be a huge variation here, so that's a ridiculous thing to Google because it depends on the strain of bacteria and that. But there we go. For many common bacteria, the generation time is quite short, in the range of 20 to 60. For most common pathogens in the body, they're closer to 5 to 10. So let's say that we were 5 to 10 hours, sorry. So but even still, let's make this 20 minutes, right? Just on the sake of argument. Right? That still means we'd have 1 billion in a mere 260 minutes. And how long is 260 minutes?
Whoa, 2 hours is 120.
2 hours and 20 minutes?
Yeah, right? Pretty darn quick. Now, will they grow indefinitely? No, of course. Eventually they'll run out of food, right? But now let's say it's even 6 hours, right? Okay, so let's say it takes 6 hours, which was the upper limit there. Even still, here we are at, here we hit 1 trillion, and we would have 1 trillion bacteria after 306 hours. And what's 306 hours? A mere 12 days. So you'd have even at the longest span they gave for most bacteria, you'd be talking about having 1 trillion of them by under 2 weeks. Bacteria reproduction happens fast. Because again, once you go from 1 to 2, both of those can reproduce. Once you go from 2 to 4, all 4 of them can reproduce.